MALDI is used in matrix assisted laser desorption ionization. So there is a laser. So laser falls on your sample and sample gets ionized. So this ionization is done through different sources, and then this ionized sample travels in the mass analyzer. Okay, and gets separate in that mass analyzer. So this is MS1 analyzer. Now when you are talking about mass spectrometry, when you are talking about MS only, so in that MS only one mass analyzer is there, and it will produce a precursor ion, or it is also called as a parent ion, because from sample ionization is done, and this directly this ionized ions are enters into the mass peak. And when it is only MS, then after entering this, it directly goes to the detector and detects the precursor or parent ions only. But there are different nowadays, more sensitive mass specs are there, in which it is called as a tandem mass spectrometry, in which it is called as MSMS. Means there are two, this MS1 means mass analyzers are attached. Now as I have told that parent ion enter in this, okay, it gets separated, and then again the second source is there, which again analyzes this parent or precursor ion. So this is done by collision. Collision photon source is there, and again this parent ion is break down into the fragments here, and then again these fragments enter into the MS2, second mass analyzer, and again the product ion is formed. It is also called as a daughter ion, parent ion and daughter ion, and this daughter ion now get detected on the by the detector. So this is tandem mass spectrometry, MSMS, where two times the ions are going to be fragmented and detected by detector. So what is the principle of tandem mass spectrometry? First, ionization, creating ions from a sample. Mass analyzer is there, separating them by their mass to charge ratio, and detector recording the relative abundance of each ion. Okay, so this is what the principle of mass spectrometry. And nowadays tandem mass spectrometry is only used, MSMS. It gives very sensitive and very you can say robust and good results. Now there are different types of mass spectrometry. So this is LCMS analysis. See, this is we are talking about only LCMS, liquid chromatography mass spectrometry analysis. So here there are different mass specs are given. As per the mass specs evolved, first single quadrupole LCMS analysis was there, then LC time of flight MS, means to the liquid chromatography a time of flight tube is added, and which is really very good, giving very good sensitivity and identification in this. Then this FTICR MS, here the breaking of the molecule is done by different method, and orbit trap. Now orbit trap is a very, you can say, nowadays it is a very sensitive and very good mass spec for all types of omics, such as proteomics, metabolomics and lipidomics. And you can do here a targeted analysis and also a non-targeted analysis in orbit trap. Now these are the different mass specs. In this we can do the targeted analysis, which is triple quadruple LCMS analysis. This MRM is nothing but the multiple reaction monitoring, which I am going to explain now. QTOF. In this also you can do the targeted analysis, and triple TOF, the targeted analysis can be done. Now what is untargeted and targeted? Untargeted analysis is mainly done for you can say descriptive studies, or you can say the identification of biomarkers. So whole metabolome, whole proteome or whole lipidome you are going to identify in this. non-targeted. But in target analysis, if you know that yes, I'm having these three molecules which may be the biomarker for particular disease or for particular plant extract, so I want to identify and quantify only these biomarkers. So at that time you should go to the targeted analysis, in which you are telling mass spec that please identify this particular molecule from my sample and give quantification of that molecule also. So there is a workflow of targeted analysis. This workflow I am showing regarding the proteomics. The same workflow is followed for metabolomics also. So in protein, when we are going to extract the protein from sample, if it is a tissue or serum sample, in proteins albumin is very much mask the lower concentration or lower concentrated other proteins. Albumin concentration is more than 80%, 80 to 85% in blood or tissues. So you need to deplete this albumin, then only you can able to identify and see the lower concentrated proteins in your sample. So then you have to denature that proteins by using trypsin enzyme, and then you get the peptides. These peptides are gone through different alkylation and reduction method, and then they are subjected to triple quadruple machine. So this is triple quadruple machine. In this triple quadrupole machine, there are three quadrupoles, means three you can say electrodes are present, Q1, Q2 and Q3. In this, when you enter the sample in this quadrupole, the peptide selection is done. Means what? If you want a particular peptide to be identified, okay, so you say to the machine that yes, I want only this peptide sequence to be identified. So that machine allows to enter only that particular peptide in Q1, quadrupole 1. Then in quadrupole 2, this peptide enters and gets fragmented. And these fragments, again there are selected fragments of this peptide which are having higher concentration. These selected fragments with higher concentration only enters into the third quadrupole, and then this signal is reached to the detector, and detector detects that particular your specific protein peptide, and you can identify that protein, and you can quantify also that protein by using this triple quadrupole machine. So the principle remains the same for all these machines, in which you can do the quantification. It is called as multiple reaction monitoring, because you can give at a time more number of, like say 30 to 50 number of identification of proteins can be done at one time. So it is called as a multiple reaction monitoring transitions. And there should be some source or some software which can identify and quantify. So in proteomics, Skyline software is used to identify and quantification of these your targeted proteins. So this is what the targeted analysis done. The similar way it is done in metabolomics and lipidomics, but only the software is going to change. Now, untargeted metabolomics is the profiling of detectable all metabolites present in sample. Mostly it is done through Orbitrap, LCMS and QTOF. Targeted metabolomics, quantification of specific metabolite classes such as amino acids, lipids, you can say, or aldehydes, or ketones, or sugars, different sugars can be done, and this is done in triple quadruple mass spectrometry. Only these mass specs can identify the targeted metabolomes. And there is another term is called as metabolic flux analysis. In this, the labeled isotopes are can be can be given in vitro analysis, in vitro studies, and these metabolic pathways, by targeting some metabolic pathways, this can be you can study in the real time analysis in this metabolic flux analysis. Now, so overview of the omics workflow. This workflow is similar or which is same to proteome, metabolome and lipidome studies. First, the important step in this is sample preparation. So how you are going to extract the metabolites from your particular sample, which method you are using, which chemicals you are using. Mostly methanol, chloroform and water is used for extraction of metabolites from your samples. So standardization of this method for the samples is very important here in this step, because metabolome is a very versatile, it is contains a very versatile compounds with having polar molecules, non-polar molecules, and they are having steroids. So many different type of molecules are present in this metabolome. So it is very crucial and important step here only, the sample preparation, because if you are not able to extract the nearly 90 to 100% metabolites from your sample, if you are losing something, then your purpose will not be resolved. Then after sample preparation, mass spectrometry is there. So you are going to separate your compounds in this and then identify the compounds by using the mass spectrometers. And the last one is in this workflow is data analysis. So data analysis is done by using different statistical softwares here. In case of metabolomics, the Simca software is there. This is a paid software. And Metabo Analyst is another one software which is used for data analysis. And this Metabo Analyst is the free software, and mostly this software is used for analysis of the metabolomics data. Now, in detailed workflow of metabolomics: study design, question, sample size and controls. These things are going to be same for all omics. Means what? That if you want to understand the metabolome or study the metabolome of, for example, breast cancer patient, okay, you want to uh, identify the early diagnosis metabolome marker from your breast cancer patient tissues. Now here you know the question. Now, importance here important is calculate the sample size. So sample size calculation is same for all omics studies. So it is having it is having one formula, and then you can calculate the sample size. And controls also you have to take healthy controls, the patients who are not having the breast cancer. These are your healthy controls, and so you are going to design the study. Then sample collection. So sample collection is important. How you are storing the samples is important. And then extraction, as we have discussed. In case of metabolomics or metabolome extraction, mostly when you are going to the GCMS, GC means gas chromatography, mass spectrometry. Still now, till we have discussed regarding the LCMS technology. This is a very commonly used technology, LCMS. But for particular type of metabolome or metabolomic studies, gas chromatography, mass spectrometry is used, and it gives very nice results when the sample is containing more than oils, alkaloids. So in such cases, if you use gas chromatography, mass spectrometry, it will give you good results and good sensitivity. But the extraction, after extraction of metabolome, a particular derivatization step is needed here when you are using gas chromatography. But sometimes in LCMS also derivatization step is needed if your sample is having oil based or alkaloids. So this derivatization is done by adding some chemical compounds such as acetylation or salicylation. So these compounds are added to your metabolites, metabolome, and then after adding this compound to these metabolites become more stable metabolites, and they can be easily detected by the detector, and they cannot get you can say exhausted before they enter into the mass spec analyzer. Okay, so mostly gas chromatography technique needed the derivatization of your sample. Then as after extraction and derivatization, analytical platforms you can different use as per your sample and as per your question of study. So LCMS, as I have told, GCMS and QTOF. So after that, data acquisition method which you are using in your mass spec is very important. So there are two data acquisition methods: DDA, data dependent analysis, and DIA, data independent analysis. So DDA is the method if you set into your mass spec, then it will scan the sample and it will identify the mostly abundant ions are present in your sample. So what will happen here? That you will lose the low abundant molecules from your sample. So this is what the drawback of this DDA analysis. But in DIA, data independent analysis, your mass spec, what it does, it takes the particular mass range window and it scans only that mass range window, such as from 1 kd to 50 kd mass range, it will scan. Then after that 50 kd to 100 kd mass range it will scan. So in this particular acquisition method, you are going to you can say identify nearly all compounds here. it it don't see that either it is having a low quantity or high quantity ions. It will detect all ions which are coming inside the machine. So this DIE is mostly used for um, exploratory omics techniques, means for identification of biomarkers. And DDA many times people use in targeted analysis also. If you know that in your sample your biomarker is having a higher concentration, then DDA will work good. Now after acquisition of your sample, data processing is done. So this data processing, there are different search engines are present in which what it do, you are having one spectra which is your practical spectra you get from your mass spec, and this search engine searches different databases and sees that either that your practical spectra is going to match with any other spectra, theoretical spectra which is present in these different databases. It goes there and see, and if it find yes, this is the spectra related, it is resemblance 100% or 99% resemblance to my alanine amino acid, then this may be the alanine amino acid. Okay, so it picks it there from and it gives 100% confidence interval with alanine amino acid. Now further you get such type of list of your metabolites generated through your machine, and it will show you the list along with your confidence interval, how much percentage by confidence you are identifying that compound, and then it will show the very important in this is your retention time and mass of your compound. Then statistical analysis is done as per I have told by two particular softwares: MetaboAnalyst, which is a free one software and mostly it is used by many people, and Simca. Simca is the chargeable software, but this is having very good different modules of PCA, PLSDA, and you can do hierarchical clustering also in this, and you can do such as to identify which biomarkers are having with a high confidence interval or with high statistical values. So this can be done in Simca. It can be done in MetaboAnalyst also, but this works on different algorithms. Now, after that, then what you will do? After you get a list, from this list, you will cut out the list of the biomarkers or of the compounds which are highly statistically and with high fold you are going to identify. So, for example, if you get here 1000 compounds, in your statistical analysis, you will get only maybe 100 compounds are coming after your statistical analysis. Then these 100 compounds you are going to give for pathway mapping and biological interpretation. Now, this is done by different, you can say, databases, in which these are only for the pathway databases, identifying the pathways of your interested one candidates, those 100 candidates. So these are done, this is done by cake, HMDB database and Metlin. These databases only for this HMDB and Metlin is only for the metabolomics. Then the another thing you can do here, that you can identify the GNPs, means global natural products social molecular networking. So here you are identifying the chemicals in this, and this is the database for that, and this is mainly for looking out the chemicals, active ingredients which are present in your herbal formulations, or these are also called as herbal metabolites. Now further, this study you can take to integrative multiomic approach. Means if you are doing parallelly proteomics or lipidomics of the same samples, then you can integrate this all these data, or all these data integrate with that, and then see which pathways and which pathways are going to be common in all these four. Means there is a relevance between the this particular pathway and your object or your question. So this multiomic approach or multiomic study, by integrating all these multiomic you can say results, can be done by this metabo analyst. So this is what in this year 2025 they have added this particular feature in metabo analyst. Now, analytical platforms in Ayurvedic metabolomics. Which platforms you are using in here? As I have told, GCMS, gas chromatography, mass spec. So, as we know that for making the compounds in volatile nature, higher temperature is needed. And mostly these essential oils and volatiles are going to be identified by the GCMS. If you know that this particular plant is having more percent of oil in it. Okay. For example, Nilgiri plant, Nilgiri, you can say leaves. So more metabolites which are present in that Nilgiri leaf, when you are going to extract, they are having oil in nature. Okay. So if you are taking that for identification in LCMSMS, you will not get the good identification, or you will not get the 100% identification there. So for that, you are going to use only gas chromatography here. So gas chromatography will give you the 100% identification with 100% you can say sensitivity and specificity. Now for the LCMSMS, here you can identify with higher confidence levels flavonoids, alkaloids, polyphenols, and all your aldehydes, ketones, and your sugars, and all these you can identify. And proteomics work is also done very nicely in LCMS-MS analysis. This Qt of MS you can use for untargeted profile, means for targeted one, untargeted one for identification of biomarkers or for identification of certain compounds from your whole metabolome, and for identification of certain compounds from complex formulations, such as more than 10 or 12 drugs are combined together, and you want to identify and quantify the particular chemicals from that, then this Qt of MS is gives you a good result. And this UHPLC means it is nothing but the HPLC, but ultra pressure HPLC. It is that it's so before giving the sample to mass spec, you have to, you can say, fractionate the sample, so that you are reducing the complexity here. In HPLC you are fractionating your sample, or say you get 20 fractions of your sample which you have injected in HPLC, and then that one fraction, one by one, you are giving to the mass spec for identification of your compounds. So here the complexity reduces in UHPLC, and so that you can identify the 100% compounds. Now Orbitrap is a very sensitive machine, and it is very popular nowadays. It is having little bit different technique they are using in this for identification. Means for ionization, the same technique is there, ESI, but its analyzer is having the different technique. So it's become a very high resolution, high sensitive, and highly specific results you can get from this. So you can use it in untargeted and targeted in both. So Malditov, this is used for herbal fingerprinting only. FTICR-MS is ultra high resolution analysis can be done, and mostly used for untargeted analysis, and gives very good results. And NMR is used, it is not a mass spec, it is only used for structural identification of your novel compound or unknown compound. You can get a total structure of that particular metabolite or chemical compound. So GCMS, how you can use in Ayurveda research. So what is the strength of this technique is, it is having high separation efficiency and suitable for only volatile metabolites. For this purpose, volatile metabolites, we need to derivatize the sample first, and then only we have to submit to the this machine. So what is the applications? Profiling essential oils in medicinal plant. Standardization, and for standardization quality control, you can use this, so that the detailed chemical profile of Ayurvedic formulations such as oils, decoctions, and aristas you can get through GCMS. Identification of bioactive compounds, so which are responsible for therapeutic effects in Ayurvedic medicines. Understanding the therapeutic mechanisms, and research and development means you can provide the foundation for discovering new therapeutic agents and developing novel treatments. Now LCMS, this method having strength that it can detect the wide range of metabolites. It is not restricted to only volatiles. Mostly no need to do derivatization. You can directly add your sample in this. Okay, so it is having very high sensitivity. So all that Orbitrap and LCMS, MS, all these come under the LCMS technique. So Orbitrap is the most popular nowadays, and chemical profiling, the application is chemical profiling of formulations. So this allows for detailed chemical characterization of both single herb extracts and polyherbal formulations. Also used for quality control and standardization, to standardize the doses and ensuring the consistent quality of Ayurvedic products can be done by using the LCMS technique. As Orbitrap, you can use for you can exploratory research and also for targeted. So these both can be done, quality control and standardization, in Orbitrap LCMS. And scientific validation and understanding mechanisms provides the insights into the molecular mechanisms of Ayurvedic formulations to exert their therapeutic effects. Detection of undesirable compounds. If there are some toxic compounds are present in your sample, then you can detect by using LCMS and avoid the particular formulation. Identification of bioactive markers, which are essential for assessing the extract therapeutic potential, as there are some compounds which are very active against some diseases which are present in your formulation. So that active compounds percentage, how much purity is there, you can identify by using the LCMSMS. And you can do pharmacokinetic studies also to quantify the constituents of Ayurvedic formulations, biological fluids, helping to understand their absorption, distribution, metabolism, and excretion profile, and assess the overall bioavailability. And also drug discovery and development, you can use the LCMSMS. Now, this UHPLC top MS can be used for metabolite profiling and biomarker discovery. Pharmacokinetic studies can be done, and understanding the metabolic pathways you can done here. And high resolution, this is a high resolution untargeted analysis and suitable for complex mixtures. So as I have told, Orbitrap is a very gold standard method, mass spec now, for identification of herbal ingredients. It can be used for quality control and standardization. Adulterant detection such as steroids can be detected in the herbal drugs, and research and development by providing detailed chemical information, or you can identify the novel compound also. So Malditov is the another mass spec. This is you can use only for quick fingerprinting of herbal formulations. For example, if you know that in my herbal formulation, A, B, C, D are the key ingredients. So, only that you can check directly, quickly by using multi-topic mass spec, and so it is very suitable for quality assurance. And FTICRMS. So, this is Fourier transform ion cyclone resonance mass spectrometry, and this is having high resolution, identifying and quantifying bioactive compounds, assessing the authenticity and quality, and understanding the complex chemical composition of Ayurvedic formulations, and used for untargeted metabolite discovery. So what are the challenges in Ayurvedic or Ayurveda metabolomics? It is a complex multi-herbal formulation. This is a challenge to elucidate the complexity, to resolve the complexity of these formulations. High variability in plant metabolite content. So as you know that if you are growing the same plant in two different geological conditions, then the metabolic content will be variable in that, and the active ingredient which you want may be having the different percentage in different geological conditions. So these are the challenges in assessing the metabolomes. Variability and standardization. So difficulties in standardizing raw materials and manufacturing process. This can be done by using mass specs, metabolic database limitations. Now, there are many chemical compounds are present, and not we are not having the mass spectra of every chemical compounds. There are unknown chemical compounds also. So this is the limitation of database. If you don't know that compound, how that the spectra is present in your database, mass spec database. Okay, and if it is not present in the database, if your sample is having that particular metabolite, but you are not able to identify that metabolite as it is not present in the database. So this is the really challenge to identify the novel metabolites or novel chemical compounds and incorporate that novel chemical compounds into the databases. Biomarker identification. So hurdle with therapeutic efficacy and safety in complex. Inter-individual variation. Now, when you are giving some drug to a patient, then the highly variable metabolome and it is having a genetic variability, environmental variability, gut microbiota also influences on this, and so it influence on analysis and interpretation. Then chemometric tools are very less, which can help in authentication and ensuring the batch to batch consistency. Interdisciplinary collaboration is lacking here. To bridging the gap between Ayurveda and biomedical science necessitates the collaboration among researchers, practitioners, and regulatory bodies. And standardized protocols are not there for development and implementation of validated protocols for metabolic profiling, quality assessment, and clinical trials are needed to ensure the reliability and reproducibility of the results. So what can be the future directions? For scientific validation and quality control, comprehensive metabolic profiling done, a unique chemical fingerprints for quality and validation of herbal products can be done. Marker-based quality assessment can be done. For example, to identify the pharmacological active compounds and ensure the consistency in herbal medicines. Understanding the variability means analyze the factors assessing the quality of herbal products. Uh, Identifying the novel bioactive compounds for potential drug development, and mechanistic insights are needed for Ayurvedic medicines using network pharmacology and metabolomics to understand how they interact with disease-related networks, means here pathways. Now, for personalized medicine and clinical application, we can link metabolomics to Prakriti to develop personalized treatment strategies, and also clinical trials can be done to, and in which Panchakarma based Ayurvedic treatments can be applied and understand the resulting metabolic changes here. And for systems biology and technology integration means multiomics integration should be done so that we can get the holistic view of how these molecules are behaving or interacting with each other. So if I'm having time, I can quickly show the some results of my proteomics study which are related to omics only.
Yes, madam. Please proceed.
Okay. So now in proteomics is having different two types of quantification techniques, uh, gel based and gel free. As we know that polyacrylamide gel electrophoresis we do for identification of protein by using Western blot. Okay, so nothing but that is only used here, but it is two-dimensional gel-based technique, and dye is there in which we are only labeling the proteome and doing the 2D gel electrophoresis here. And another is the gel-free. So the proteins are in solution and we are labeling them in solution, and we are assessing or identifying them in mass spec in solution form only. So here this is the workflow of subtypes and HER2 overexpressed stages of subtypes and stages of breast cancer of HER2 overexpressed, which I have done in my PhD. So just to give how the method or the pipeline of proteomics, metabolomics and lipidomics you can design. Okay, pipeline is same only. Here I have done proteomics. You can do lipidomics or metabolomics also. So first is the standardization phase. The extraction of metabolome or proteome standardization is very important so that you ensure that your method is working fine and you are extracting as much as possible 100% or you can say 90% of your molecules. Then discovery phase is there. In this, you are going to identify all metabolome or all proteome present in your sample. So in the discovery phase, I use two methods, gel based and gel free method. Both are labeled one. So again, what I have done, I have taken the control, means healthy control samples from breast cancer patients, means these are tissue, but these are 5 centimeter away tissue are taken from the MRM samples of breast cancer patients, and luminal B subtype patients tissue and HER2 enriched subtype patient tissue I have taken here, and here stage 1, early stage 1 and late stage 3 samples were taken. And the same thing I have done in this gel free also. So after getting the identifying the proteins with these two techniques, I have further done the statistical analysis of those proteins to identify the highly significant proteins present in my sample and which are having very high fold, more than two-fold expression should be there. So I have done first BB and EDA analysis through so in my Daiichi techniques, the software is comes along with it, which is called as decider software. In that I can do the BB EDA analysis, marker selection with PLSDA tool, and then again uh, I have done by Simca also all these things. And in this gel based, now you have to identify these proteins after you are doing all the statistical analysis. Okay, but in gel free uh, or in liquid, you are going to do all statistical analysis after identification. So here, once I get the list of those proteins which are highly significant, I have submitted that list to the pathway and network analysis using different bioinformatic tools. So here, Panther, David, WebGestalt and String I have used. String gives the protein-protein interaction here. So I found the different pathways which are acting or which are playing a role in that particular either stage 1 or what are not present in stage 3. So what pathways are present in stage 1? I got most of the proteins which are expressing in Wnt signaling pathway. So this pathway is very active during the initial stages or initial or you can say that during the inception of the cancer. And the Wnt signaling pathway is also um, became active in during EMT late stages such as T4, and when the cancer spread is there, na, those you can say stem cells are going to migrate and they are going to establish them in another position, another part of your body. At that time also EMT when goes on, at that time also Wnt signaling pathway was very active. So these things can be known by using this different pathway analysis. And then after doing this, the proteins which you are going to find as a biomarker, for example, I have got the 6 proteins which are expressing in stage 1. So these proteins again I have to validate. So for that I have to do Western blot of those 6 proteins only and multiple targeted analysis by using mass spec, multiple reaction monitoring. So in case of metabolomics also you have to validate your metabolome if you are doing the discovery, you are doing the biomarker discovery. So in that you can't do Western blot of metabolome or lipidome, but you can do the multiple reaction monitoring by using the targeted analysis mass spec, triple quadruple mass spec. Now I will skip this iTrack workflow. Here, then after getting me the identified proteins, I have submitted it to the Simca software, and then I got such PLSDA image. So in this you can see that this blue is a control, this green is a benign breast sample, this is a grade 1 sample, and this yellow is the grade 2 samples patients. So you can see that these hubs or these are clusters which are going to be separated from each other. They are not they are not going to be mixed with each other, you know. So from this we can show that yes, our sample is having the variability in between different control benign grade 1 and grade 2. Okay, then I have done this to show the that how these samples are different from each other, and then done the hierarchical clustering to show that yes, my grade 2 is present in another hierarchy, grade 1 is present in another, benign is in not in that hierarchy, and my control is also going in the another hierarchy. So all are different from each other, means my whatever samples I have taken, if I have say taken the fake samples for example, this such image will not get. Okay, you will get the mixed samples here. So this will also show that yes, whatever you have taken the samples, whatever study you are doing, that is the authenticate study. Mm, and you are proving it by your statistics. Then this is called as volcano plot. This I have plotted whatever the 100 and 150 proteins I have identified through significance, statistical significance, I plotted in this. And the proteins, these are the protein spots. So these are the proteins which are present in the upper side of this volcano plot. Now these are you can say very highly significant proteins which can be used as a biomarker. So here these are negatively regulated proteins, and these are here positively regulated, means positive fold, you can say the two-fold upregulation, here 2.5 upregulation, and here two-fold downregulation, downregulated and upregulated. So further, the proteins which I got through my volcano plot, that proteins further I have used to identify the you can say the protein-protein interactions or network interaction analysis. It is also called as gene ontology. This is done in string. So if you can see here, Pantherite David, these are the databases which give you the in which pathway, which pathways are involved specifically in your this subset of proteins. Grade 1 specific proteins, I found these pathways are mainly involved. P53 pathway, this gives the aggressive nature of the breast cancer cells. P13 kinase, Wnt pathway, which is present from inception only. And these FAS and FGF, these are growth related pathways. So you can say the growth of the cells needed here. So all these pathways are coming in my grade 1 specific proteins. So I have done the string, means protein-protein interaction, and what I could found that in grade 1 proteins, I found the three different clusters are there. And if I found which these clustered proteins in which pathway they are active, so the first cluster is active in morphogenesis and proliferation of cells. The another second cluster is involved in protein folding, because folding is needed here. As the proliferation is going on, you need that more number of proteins should be you can say synthesized, and functional proteins should be synthesized. So folding is needed. So protein folding genes are active here. And in third, cell communication. So to invade or to proliferate, one cell should communicate with the another cell, you know. So for that, these genes are also highly involved, in which they are showing the cell to cell communication to proliferate the cells. So this proteomics I have given only the example how you can analyze or identify which pathways are involved, how you can interpret your results, and how you can show that your study is giving you something novel, that for example some metabolites are involved in particular pathway. So this pathway if you can if there is one particular active compound which is switching on or switching off this pathway of your drug, then you can say that yes, this pathway is switched on in the disease, and now because of my drug this is switching off, and because of that the cancer cells are dying, they are not proliferating. So it is very important to show where your drug is acting actually. So these things also you can do by using these omic studies. So thank you for listening me patiently, and this is what my presentation for today. You can ask me the questions.
Participants are hereby requested that if they have any doubts, they can just type it in the chat box. Madam, there are few questions in the chat box. We'll just go through it.
Yeah. Wait for a minute. I'm not able to.
Just a moment, ma'am. Just a moment. We will read for you.
Okay. Chat box on my screen.
Madam, there is one question by Dr. Rohit Johari.
Yes.
The question is, herbal fingerprinting includes.
Hmm. Uh, Herbal fingerprinting means if you want to identify some chemical compounds which are present in your formulation, then you can directly identify those by putting your formulation in mass spec, means but that your sample in your mass spec, and you can identify either that active compounds are present or not in that particular case.
Are there any more questions? Then you can please type in the chat box to the participants. This PPT will be shared in the Google Classroom, so it will be available for everyone to just go through. The next question is, ma'am, diabetic patient, if we collect the blood samples.
Hmm?
Diabetic patient, if we collect the blood samples. Second, है ना, to know the metabolites to differentiate the diabetic type. Is it possible? The
Yes,
next
it
question.
Yes, it is possible. Only you have to compare that sample with the healthy control persons. No, so you can do comparative study here. So you have to submit or you have to identify if the metabolites present in healthy control and also in diabetic patient. Then you are going to compare that which metabolites of, for example, if there is a particular metabolite A, it is very high fold, significantly highly increased or upregulated in diabetic patient, but not in your healthy control. That means that is a particular marker for diabetic. That may be this particular marker for diabetic. So such type of study you can do.
Are there any questions?
The full form of LCMS is liquid chromatography mass spectrometry. Pardon?
Madam, hello.
Hello. Yes, sir.
Madam, can you share a few resources with the participants that if they want to learn more about this, what they can do, or which kind of books or any sites or anything to be
Yes,
referred?
I can share it. I will send it to Dr. Shukla, or on right now it is not possible for
No,
me,
no, no. You
but
can share. You can share to
yeah.
the same mail.
Yeah, I
as a
will
reply,
mail it.
huh?
Yeah,
As a
yeah, I
reply
will
to the mail.
reply to the mail. Yes.
Supriya madam is also here.
Yeah.
Dr. Supriya madam, are you here?
Can you hear me?
Yes, ma'am. Go ahead.
Yes. So, what I was thinking, Mohan sir, that I can see that there are many teachers, faculty members also have joined for this particular lecture series. And the experts which we will have may not like have the complete insights into Ayurveda. Now, Namita has covered the herbal part, but as a Samhita Siddhant persons, we have to think like how we can use this metabolomics to study different concepts in our samhitas. For example, if we can, like, if we have a concept, if we want to study a concept of kled or am or abhishand, so we can diagnose patients clinically that there is a samata in the body, and we can have another group of people or individuals who do not have samata in them. And if we do the metabolomics or proteomics of these two groups, then we will understand that how ama is expressed or what exactly ama means, which metabolites are increased or decreased in samata. So this is like application of metabolomics for us. Similarly, for clade-like conditions, we can also use metabolomics or proteomics to study before and after treatment effects like Vaman, Virechan, Panchakarma, or any treatment. We can study like the samples before treatment, and then we can compare it with after treatment, at what are the changes in the metabolomic or proteomic or lipidomic pattern after giving these treatments. So we have to think on a wider level, like how we can use, because the faculty members will give us insights about the overall technique. They will help us, they will tell us some applications of it, but it largely depends on us that how we are going to use it, use these techniques in our studies and apply it on a wider level. Now, one more question which nobody asked to Dr. Namita, and that is very important question, which is like cost of metabolomics or proteomics. So if she can throw some light on this aspect, that will be like really insightful, because whenever we listen to new things, we feel like, okay, we will do this for our next study. But is it possible? And which are the institutes where we can go for doing metabolomics or proteomics studies? And how much is a cost per sample? So if you can tell us, and she gave us example of healthy versus control, but we can consider different conditions according to our principles or our hypothesis, and then we can study these kind of things. And we should discuss about it more as a group of Samhita Siddhanta enthusiasts, and we should take it to more applications for our principles. But meanwhile, Namita, if you can answer
Yes.
cost and institutes where these kind of facilities will be available, and if I go with my sample, what information do I need to give to those people? Like if I go with a sample, what questions I will be likely asked, and how I should be prepared about the answers, and what would be the cost of these techniques?
Okay. Um, for metabolomics can be done in IIT Bombay, and they offer a very cheap cost with we compare with other like ICER or NCL or NCCS. Uh, Mostly it's around 2,500 to 3,000 for per sample they are taking for metabolomics and lipidomics. When we are talking about proteomics, it is little bit costly, um, because it needs, you can say, you are only going to uh, extract and give the proteins to the person,
Uh,
and they are going to
Dr.
Uh,
Namita, I would just
ah,
like to clarify that being an Ayurvedic physicians, clinicians, we do not have the facility. Maybe we will, some of us may have, some of us may not have the facility of even isolating something from the sample. So we have just blood sample. Let us assume this. And we want to do the analysis, or we have a tissue sample. So how the things happen?
Extraction is not a costly affair. Okay, you are going to extract metabolome, then only three chemicals are mostly needed: methanol, chloroform, and water. So for metabolome and lipidome, extraction is not a costly affair. Only the cost of you have to bear with LCMS grade chemicals. That thing. But for proteomics, when you are going, cost comes. All kits, all reagents there, you have to use different enzymes which should not degrade your proteome. So all these things are very costly in case of proteomes. You if you are going to labeling, then the kits are needed. Even a single kit is having 2 lakhs price. Only 9 reactions can be done. So proteomics is a costly affair. And as we all know that mass specs are very costly, and maintenance of mass spec is also a costly affair. Um, I can say that when you are comparing proteomics with metabolomics or lipidomics, metabolomics and lipidomics is a cheaper way, and you can get the samples done. You can give samples on room temperature because you are drying them. This is not in case of proteins. So you can send samples to IIT Bombay from Delhi also through courier. So this can be reduce your cost in metabolomics and lipidomics here. Um Only the thing is, if come to the derivatization, then it needs some expertise there. So if you don't have a mass spec, then you can't check that either my sample is derivatized or not well, or my whatever the technique I am using for extraction, either I am getting 100% metabolome or not. So these things you can't check there. Okay, so if you submitted the sample to any institute and you are getting only, you can say, 1,000 or 2,000 metabolites, this is very less number. So you have to there such a good rapport with that institute. That institute should tell that these are very less number, you should do or work something on your extraction method, or you have to increase the sample size, like that. So these things are there in this. So
Yes.
it is a
Yeah.
costly
Thank you.
affair. Hmm.
And one more thing, just to give an example, last thing, that in Dr. Namita's work only, she is in my institute only, so we work closely. So she, in her PhD, she has worked on breast cancer. So we all know that now I am talking purely from a Samhita perspective or Siddhant perspective, that stanya, breast cancers related to stanya, stana, rasadhatu. Rasadhatu's upadhatu is stanya, and breast cancer is somewhere related to that. And she has found that there are some androgen related proteins she
Yes.
found in breast cancer samples. So then I thought, from Samhita perspective, this analysis gave me a point to think that in Rasadhatu related disease, are we finding something which are related to Shukra Dhatu? And whether these two dhatus are related to each other then, because they are on the two extremities, first dhatu and seventh dhatu. So are there, and if we see the characters of those two dhatus, there are some similarities also. So is it like something, some relation between these two? And she has presented this particular paper in World Ayurveda Congress. So this is how we can use applications and the results which we get from metabolomics, proteomics, to you to what we say, no, upabruhan of our principles and concepts in Ayurveda. And that is what we as a uh, scholars and faculty members of Samhita Siddhanta department should be doing. So thank you, Mohan sir.
Thank you, Doctor. Thank you, Supriya Madam. And probably audience, those who are gathered here, they may be thinking, initially looking at the curriculum, everybody thought that why this part is coming in Samhitas. So probably Dr. Supriya ma'am's answer is something that will make us think that it will explore new ways. We can explore the new ways. We can think about the concepts that we want to explore in a uh, appropriate way. So, Supriya ma'am, can this explore something like Gatatva concept or Leenatva concept or any other concept? Like you said, Kleda, it can be explored, Ama can be explored. So, can we just
So
try?
I would no, in this case, I would suggest like we have to have a working hypothesis that what exactly means when we will work on Ayurvedic principle, we should have some working or operational hypothesis that what exactly we want to study and what we exactly want to get. And if we can have a working hypothesis that metabolomics can give answer even to Ashaya Pakarshagati or Gatatva or Leenatva, then we can definitely study. Leenatva, I think we can think of because in some tissue localization of some other molecules are happening, so we can definitely think of such possibilities, but for that we have to have a working hypothesis, and this is what exactly we should discuss, and we should come up with such hypothesis also, like what can be done. And for such kind of concept and such kind of research, we definitely look towards All India Institute of Ayurveda, Goa. So Mohan, so the onus is on you now.
Yes, ma'am. We are trying to explore the ideas first, generating ideas, and then probably then we can think of, as you said. So this is our effort. Now the next step, I want all the members, those who are here and those who are not here, those who will be listening this particular telecast afterwards on YouTube. If you are here, join to the our Google Classroom, and in Google Classroom there will be a discussion forum where you can write your question. So here the questions are there, but they will vanish just after the closure of this meeting. But something which you are going to write in Google Classroom will stay with you and stay with all. So probably we can have more discussions. We can use that part as a discussion board, and on board there will be visiting new faculties, and Supriya ma'am, of course, ma'am will be there, and many others will come, and then probably we can think that how to use this new concept and are they really useful. If we are at all including that as a part of our MD or PhD project or even other kind of projects, when to think of, where to start, and how to hypothesize. As ma'am said, we have to have a concrete conceptual understanding, and probably those who are new, those who are listening to this concept first time, probably for them this will be a beginning, and from here we can explore in a better way. So thank you Supriya ma'am, and thank you ma'am. I request Dr. Vikrant sir to continue with the thanks.
Thank you, Dr. Monjoshi sir. First of all, I am very thankful to our director sir, Professor Dr. Prajapati sir, Dean Madam, Professor Dr. Sujata Kadam Madam, our Associate Dean and Head of Department of Samhita, Professor Dr. Mohan Joshi sir, for guiding and supporting us to organize this online lecture series. I do take this opportunity to thank a very important person, Dr. Supriya Bhalerao Madam, who has been in continuous contact with us to design and to introduce the speakers, and even now explaining the integration part. Thank you, ma'am. I thank Dr. Namita Pendharkar Madam to guide us with this untouched and unexplored topic to the people of Ayurveda. Madam has explained about the metabolomics, their types, their analytical techniques, analytical platforms, and application of the same in Ayurveda. That too in very understandable and easy way. Once again, thank you, ma'am. Last but not the least, I thank all the participants who have attended today's lecture with the utter concentration. Tomorrow, we will be having a lecture on human gut microbiome at 7 p.m. sharp, and I request you all to attend the same. Thank you, one and all.
I want to thank all of you for giving me opportunity to talk on this topic. Director sir, Dean sir, and Mohan sir. And also I want to thank Dr. Supriya ma'am. She suggested my name to give this topic, and I can able to present my topic in front of you. Thank you, ma'am, for giving me this opportunity. Thank you. Should I end the session?
Yes, madam.
Okay. Thank you, sir.